বিধান স্যার স্টার্ট করছি।
হ্যাঁ শুরু করুন।
Good morning to all. We are today we are at the third day of our DBT sponsored summer training school, and today we have among us Dr. M.D. Mustafizur Rahman as our invited speaker. He will talk about development. Before starting his lecture, I would like to give a brief introduction of Dr. Rahman. Dr. Rahman has completed his BSc Honours in Zoology in the year 2004 from Krishnanath College, University of Kolkata. He has Zoology in the year 2006 from University of Kolkata. And he has completed his PhD from University of Kolkata at the year 2020. He has qualified NET examination for 2 times in the year of 2008 and 2009. He has also qualified GATE exam twice also in the year 2009.
You don't need to mention that. Please give the experiences and that will be enough.
Okay. He has a very long years of experience. He has worked as visiting faculty, guest faculty in the various colleges like Behrampur Girls College, Krishnanath College, Madhupur Raja Samanta Vidyapith, and presently he is working as assistant professor in zoology in Ashutosh College and visiting faculty at the Department of Biological Science, Alia University, and till date. So it is over to now Dr. Rahman. Please start your lecture.
Thank you, Shoma Kormokar, for introducing me, Dr. Shoma. And without further, and thanks other members of the organizers and for allowing me to actually giving me the honor to take part in this summer training program. And today we are going to actually learn about differential gene expression, that is gene expression that has been included in your developmental biology syllabus. And we shall focus on gene regulation that will involve development with one example. So it's a topic that will actually cover lots of things. For example, it will you need to have some basic idea about what is gene regulation. At the same time, you need to know about development. At the same time, you have to know about some molecular biology and physiology, some things. So I have divided the lectures accordingly. So without further delay, let's start it. And for that, I'm going to present it. And I don't think my face is required for the lecture, so I'm turning off my video. So let's move on please, and present the PPT. Now, please someone let me know, is it visible or not? If it is visible,
yes, it is visible.
Thank. So let's move on. So what we are going to actually study today, in broader term, it may be considered as differential gene expression, but we shall consider environmental regulation of gene expression, particularly with regard to specific genes that is called distal-less genes, which actually helps in formation of eye spot in butterfly, known as bicyclosan inana. But before we actually discuss how actually this thing occurs, we need to have some basic idea. For example, what actually the differential gene expression is. So what we know, our actually life begins from a single cell, that is zygote. And all the cells are derived from the same stem cell, that is the zygote. for ranging from say osteocyte, neurons, muscles, fats. And if you look closely, then you will find each and every cell contains same genetic complement. All of them have same complement. Why they have, except this one, by mistake, I have actually placed that as well here, and that should be removed. The sperm has half of the genetic complement, so it should not be included here. So the rest of these cells, all of these cells, say muscle cells, nerve cells, all of these cells, or the cells that develop from progenitors, for example, blood cells don't have their nucleus, but their progenitors have. All of these cells have same genetic complement because they are actually derived from, all of their have been actually originated from the same blastocyst and through the process of mitosis. And we know that in case of mitosis, all the daughter cells receive same genetic complement of their mother. So all have the same genetic complement. The problem is they look so different. They look drastically different. For example, the nerve cell is totally different from egg cell. We should exclude that as well. So nerve cell is totally different from fat cell, and fat cell is different from muscle cell. Even muscle cells actually vary in their structure as well as function. Now, how this is possible? All of them have same genetic complements. That means all of them have same genes. Then why they look so different? What is the answer? The answer is, though all the cells ranging from the epithelial cells, muscle cells, all the cells have same genetic component, but in not all cells, not all the genes are being expressed. What does it mean? In some cells, some genes are active. In other cell, that gene is inactive, and vice versa. So not all genes are active at all the times in all the cells. And this difference in expression, in some cells it is being expressed, in some cells it is not being expressed, despite being the cells of same individual and derived from same stem cell, that is the zygote. And this variation is known as a differential gene expression. I hope you got the point. If you have any problem at any point, you are always free to actually unmute and ask, sir, I have not understood the problem, because it's a summer training program and my job is to actually need to understand the topic. Okay, so don't feel shy, always unmute and ask if there is any problem. So let us move on to the next slide. So we have more or less a very brief idea about what differential gene expression is. Now, how this level of gene expression, differences in gene expression is achieved, how it is possible to actually express some genes in one cell and actually switch off the same gene in other cells. How we achieve, what are the underlying mechanisms? We have actually 4 levels of gene expression in which we actually regulated these gene expression. What are those levels? Number 1 is at the level of transcription. We know that genes are expressed, or not all genes are actually expressed in the form of protein, but in mostly those actually express, actually at first they undergo transcription, and after transcription they actually undergo translation, and after translation they form a protein. And while gene expression, and this thing is known as, this line is known as the gene expression, that means from gene to protein. Not in every case, but in most cases it is known as the gene expression. Now, so we have regulation at all the levels. For example, at the level of transcription, and this is known as differential gene transcription. The first one, the blue one on the top, is at the level of transcription. That means not all genes will be transcribed at same rate or at all cells. In some cells, some gene might get transcribed. In other cells, they might not get transcribed. Even if it gets transcribed, its rate may be different. For example, in some cell the rate may be slow, in other cell the rate will be fast, and this is known as differential gene transcription. And now next level is translation, and translation occurs through mRNA, and the regulation occurs through a differential processing of mRNA. For example, the mRNA is composed of several say introns and exons, and in many cases some exons are spliced off and some exons are included. And through shuffling of these exons, actually new type of proteins could be produced, and thus effect could be modified, that is expression could be modified. So second level is at the level of selective mRNA processing, or you may also call it exon shuffling, through which is observed in say immunoglobulin genes or something. In many other cases, not only immunoglobulins. The third level is a selective mRNA turnover stability. Now, even if the genes get transcribed and transcription rate is same, it may differ at splicing level. For example, some exons may be removed in some cases, and in some cells, all the exons may be kept, or different combination of exons may be actually retained. Even if these two are same. Even after that, actually regulation could occur, and that is at the level of turnover. For example, not all mRNAs are equally stable. Some mRNA degrade faster, and some mRNA last very long. For example, you know that mRNA that is stored in the cytoplasm of egg, then their stability is very high. Whereas mRNA that are actually should be specific. I don't have a specific example right now. That are synthesized from the nucleus and comes to the cytoplasm after their use, they normally get degraded. So not all mRNA are actually have same stability. As they have different stability, they have say different time to actually say express themselves, and the level of expression may be changed. For example, if the mRNA is very unstable in one cell, so it got very little time to actually transcribe itself, and it will have very small amount of protein. And if it's actually it's very stable, and then it will have a very long time, and it may produce actually a higher amount of protein, so expression may even change at the level. For example, let that the level of expression is the same even at that level, that is the level of RNA turnover. Even that after fourth level, expression could be modified. For example, two cells, all the three criteria are same, so they have produced same amount of protein. Even one cell may express the effect of that is express that gene, and another cell might not. How this is possible? And this is possible through modification of protein. For example, you know the kinases and the phosphorylase. What they do, if you have some idea about enzyme, how enzymes are activated and deactivated, then you know in many cases phosphorylation of some enzymes cause activation of that enzyme, and dephosphorylation of that enzyme actually regulate inhibit the activation. So fourth level is modification of the protein. So even the protein is same, in some cell it may get activated by phosphorylation or by some other means, and in other cells they may get dephosphorylated and or inhibited by some other means. Okay, so we have four levels of through which actually gene expression actually is regulated. So got the point? If you got the point, no problem, I should move to the next slide. If you have any problem, you're free to ask. Okay, unmute yourself and you may ask. Okay, I'm moving to the next slide. So we shall focus on the first type. It is, you know that this topic is very large, and they demand separate classes for themselves. Okay, so for our today's topic, we shall focus on the first level, which is the most important one at the level of transcription. So let us see how differential gene transcription is achieved. Actually, differential gene expression can be achieved by actually three means. One is by regulatory transcription factor. Just listen to them. Don't have to worry. And if you want, you may also actually copy them in your notebook. That will help you to focus. Otherwise, you may get distracted. The first thing is regulatory transcription factor. Second thing, change in chromosome structure. That is chromatin remodeling, histone variants, or histone through histone modification. And third is insulators. So we have three mechanisms by which we can actually regulate the gene expression at the level of transcription. So in some cases, through these mechanisms, we can activate some expression of some genes, and actually in other cases, we may actually inhibit the expression of things. Let us discuss about the first one, the regulatory transcription factors. Now, what are regulatory transcription factors? Now, how they actually operate? Now, you have to remember that we have, I think you know about GNL transcription factor. Can anyone of you unmute and actually give one example of GNL transcription factor? কেউ একজন কি আনমিউট করে আমাকে বলতে পারবে যে GNL transcription factor কাকে বলে? Have you gone through GNL transcription factor in your undergraduate courses or not? আছে কি তোমাদের আইডিয়া?
So example বলবো or মানে define করব? আচ্ছা,
ফার্স্ট ডে তুমি তোমার নাম বলবে, কলেজ বলবে, তারপর তুমি if you want you may actually define that as well.
Okay, so Shriparna Chattopadhyay from Ashutosh College. General transcription factors, they are the basal transcription factors, and they are like a class of protein transcription factors that bind to certain specific sites of the DNA, rather the gene that is a promoter region, to active the transcription of genetic information.
Okay, so lots. Give one example, please. একটা এক্সাম্পল দাও।
Sir.
মনে আছে? No problem, আমি দিয়ে দিচ্ছি। So what are general transcription factors? General transcription factors are factors that are mostly required in case of eukaryotic genes for basal level of transcription. For example, if you want to actually transcribe something, you need to have say RNA polymerase, and RNA polymerase will see it at the promoter. Now this seating process is not simple in case of eukaryote, and actually the RNA polymerase actually don't have sufficient affinity to bind and actually to carry on the initiate the transcription process. For that, additional factors are required, and these additional factors are known as transcription factors, particularly general transcription factor that will allow binding of the RNA polymerase to the say promoter. So what they do, general transcription, they help the RNA pol to bind to the promoter and thereby activate the transcription. And example, there are lots: TF2D, TF2B, E, H, and I think you have gone through your undergraduate syllabus and you already have some idea about what general transcription factors are. So what they do, they regulate basal level of transcription. If you add say a mRNA and its RNA pol and the general transcription factor, it will actually start synthesizing the RNA at a specific rate, which is very slow. Now, if you want to actually regulate this transcription, if you want to increase the speed or decrease the speed of the transcription, you will be needing additional proteins, which are also transcription factors, because they regulate transcription. And these are transcription factors are known as a regulatory transcription factor. So we have two types of transcription factors. One are general transcription factors that regulate basal level of transcription of every gene. Regulatory transcription regulate rate of transcription of a particular specific target gene. So we know about a general transcription factor now and the regulatory transcription factor. Now, how they actually operate, or how many types of general transcription factors are there? Regulatory transcription factors. So we have two types of transcription factor: a regulatory transcription factor and the say general transcription factor. So this is the transcription factors in the top line. We have the general transcription factor as well as we have the regulatory transcription factor. Okay. So where they bind? They bind to a specific sequence of the DNA, and that sequence of the DNA is known as a regulatory sequence or control element or regulatory sequence. Okay. They have other names also. So this sequence is in general will be known as a regulatory sequence or regulatory element or control element. So we have three names for the same thing: regulatory element, control element, or regulatory sequence. So these are the general transcription factors that will bind to this site, that is the regulatory elements. Now, based on the nature of these general transcription factors, they could be divided into two categories. So let's again move. So we have regulatory transcription factor. Transcription factor can be two types: general and regulatory. And now we are looking at the regulatory transcription factor. Regulatory transcription factor can be of two types. One are activator, and second one is suppressor. Which are activators? That actually activator increase the rate of transcription will be known as activators. And repressor, the genes that will actually repress the expression of that gene will be known as a repressor. That means if this one, if this one actually is general transcription factor, if it actually increases the rate of the transcription or initiate the rate of the transcription, then it will be known as activator. That's why it has been actually marked as activator proteins. And the sequence, which is known as regulatory sequence, to which this activator proteins now will be called enhancers. So it's a specific name. So all of them, maybe you are student of MOOC BDA Godadhar College, but some of you are say third year student, some are second year and first year. So like all of them are, both of them are actually regulatory elements, but those that bind with activators will be known as enhancer because they enhance the rate of transcription. And those actually that will bind to repressor and repress or inhibit the transcription will be known as silencer. So activators will bind to enhancer and repressors will bind to silencer. Okay. Now, both activator and repressors are regulatory transcription factors, and both enhancer and silencers are the sites to which these factors actually bind. Now, enhancer binding at activator increases the transcription rate, a silencer binding to the repressor replaces the transcription rate. I think you got the point. If any of you have any confusion, you are always free to ask. I'm giving you a few moments. Okay, so I think you got the point. So let's move on to the next thing. That is how these actually enhancers and suppressors actually work. How they actually increase the rate of the transcription. There are several mechanisms of doing this, and three of them actually have been shown here. One is through hormone binding. We shall discuss them later on. And second one is through protein-protein interaction. And third one is for modification of phosphorylation and modification of these transcription factors. কেউ কিছু বলছো? So we through three mechanisms. So let's discuss the first mechanism, how they actually activate genes through this mechanism, that is through hormone and receptor binding to the small molecule to the regulatory factors. So all of these factors, the transcription factors, actually have three domains. Okay, so regulatory transcription domain, we have three domains. All of them have, let's change the color, please. So let's suppose this one is a regulatory transcription factor. Now all the regulatory transcription factor will have three domains. One domain is the DNA binding domain. Through which it will bind to these sequences, or they will bind to the DNS. That's why they need this one. A second one is a site for effector molecule binding. And third is a site for covalent modification. So all general transcription factors, regulatory transcription factors, all regulatory transcription factors will have three sites: DNA binding domain, a DNA binding domain for say binding of activator molecules, and third is site for covalent modification. So let's clear that. So how they actually modified. One example is we are going to actually use this one for our purpose. So let's have another example for steroid hormones. Steroid hormones receptors are cytoplasmic receptor, and sometimes they are nuclear receptor. So what they do, steroid hormones, they actually diffuse inside the cell and they bind to these receptors, which are actually regulatory transcription factor. After binding, they undergo dimerization, and these dimers actually get inside the cell and they bind to the enhancer or a regulatory DNA sequence, and thereby activate the expression of desired gene. So this is the first mechanism. And how they do it, actually we don't have enough space for discussing those things. They actually form a hairpin-like structure, and they actually recruit say modulators, and they actually associate with the RNA polymerase, and thereby actually increase or decrease the rate of the transcription. So this is one mechanism. We have studied it a little bit in detail because we are going to use it, and you should try to remember that. And now second type of modification is they do the other things the same. In this case, actually the activation occurs or repression occurs through protein-protein modification. That means two of the say regulatory transcription factor, they actually bind to one another to form a homodimer, and this homodimer then binds to the regulatory element. In this case, it is enhancer if it enhances the rate of the say reaction, and this homodimer formation then again will recruit the modulators and actually activate the transcription of the gene through activation of the RNA polymerase. So another mechanism is, you already know about that in case of G-protein-coupled receptor say transcription. So that is through phosphorylation. So in your say G-protein signaling classes, you have learned that G-protein-coupled receptor activates adenylate cyclase, which increases cyclic AMP. Cyclic AMP then binds to regulatory subunits of protein kinase A, that is PKA, and by this binding actually frees the protein kinase A, and these protein kinase A then get translocated to the nucleus, and where they actually phosphorylate the CRE binding protein, and this CRE binding protein is actually a regulatory transcription factor. And after binding and phosphorylation, actually phosphorylation of this CREB, that is which is actually a regulatory transcription factor, transcription start, if you can recall. So these are the three basic mechanisms through which actually the regulatory transcription factors work. Okay, so hopefully you got the point. And if you have any problem up to this point, you are again free to ask. Otherwise, I shall move to the next slide. Don't be shy. If you have any problem, you're always free to ask. That's why I'm here. So I think you don't have any problem. So let's move on. Okay. So there are other change mechanisms through which actually we can get this type of modification. Actually, we got 3 types of modification. One is through regulatory transcription factors, and second one was change in chromosome structure. And change in chromosome structure, I'm not going to actually discuss it in detail. In this case, what we actually do, and normally we actually modify the histone and thereby actually change its place. For example, it could have a 2 conformation. One is called open conformation, and another is closed. Actually, the DNA is not freely accessible for the translation, transcription. So if there is DNA and this is the promoter region, now this RNA all binds to it and transcription occurs. It happens very easy, and it's easy in case of prokaryotes. But in case of eukaryote, this thing is not that simple, because DNA is not naked. It's not freely available for actually transcription. What is the problem? They actually remain associated with histones. And this histone packaging, so now the DNA is wrapped around a solid body, which is histone. Actually, this wrapping around actually reduces the access of this promoter to say RNA polymerase. Not only that, it actually reduces entire access of the entire gene. So now the RNA polymerase will not be able to bind with the promoter and transcribe the gene easily, because it is wrapped around the histone. So this conformation could be changed. If it is very tightly wound, then it is very difficult for transcription to occur. And this conformation is known as closed conformation. And this conformation could be changed in some cells and could be kept tight in other cells, through which actually modification could occur. And this is known as through change in chromosome structure. So how it is achieved for actually say sliding the histone through the DNA, so that you get some open portion or free DNA to access the DNA and start the transcription, or keeping it tight as possible. One thing through which you can actually achieve say access to the DNA and differential gene expression through this type of modification. And second type of modification is through removal of histones. So if you remove histone from a portion of the DNA, so if you remove these 2 histones in particular, then that sequence region of the DNA will be free, and that conformation is the open conformation and will allow the transcription to occur. And if you actually keep it wound around the histone, then that will be the closed conformation. So by removing histone in this case, or sliding the histones through the DNA, we can actually close or open the DNA access for transcription to occur, and thereby we can change the rate of transcription or change the transcription altogether. In some case you may switch on, or in some case you may switch off the rate of transcription. And third thing is actually using histone variants. And histone variant means actually in case of eukaryotes there is more than 120, as far as I can recall, a type of histone variation, and we have 70 genes, separated genes for synthesis of histones. Most commonly the histones are, I think you already know, these are H1, H2A and H2B, and H2B, H3 and H4. These are the most common variants. But beside that, we have some other variants, and some of the variants, presence of these variants actually increase the rate of transcription or chance of transcription, and in some cases it will decrease the chance of transcription of the respective gene that remains wound around that histone. কোনো জিন যদি একটা স্পেসিফিক ডিএনএ সেই হিস্টোন প্রচুর দিকে জড়ানো থাকে, because of the nature of that histone variant, it may decrease or increase its transcription rate. For example, H3.3 is a histone variant that actually stimulates the rate of transcription, as far as I can remember, whereas H10 reduces, it enhances the rate of transcription and it decreases the rate of the transcription. So based on the presence of say histone variants, in some cases say presence of H3, if the gene is wound around as H3.3 instead of H3, so it will have increased probability of getting transcribed. And in some other cell, if it is actually wound around say H1, which the place is different, instead of say H10 instead of H1, then it will have less probability of being expressed. Same genes, so because of presence of different histone varieties, they will be expressed at different rates. Okay, so hopefully you got the point how structural modification actually can lead to say change in gene transcription. If you have any confusion, you're again free to ask, and I'm waiting for to hear from you. Okay, don't be shy, unmute and ask. Okay,
sir.
হ্যাঁ বলো, তুমি কে
Sir,
বলছো,
বলছি।
কোন কলেজ থেকে বলছো?
স্যার আমি সার্থক মুখার্জি, বিকেসি কলেজ।
আচ্ছা বলো।
স্যার এই যে মানে হিস্টোনের যে নিউক্লিওসোমের পজিশনটা চেঞ্জ হলো বা যে অক্টামারটা রিমুভড হয়ে গেল, এগুলো কি কোনো সিগন্যালিং এর থ্রুতে হয় নাকি মানে মানে কিভাবে হয়, মানে ওরা বোঝে কি করে যে ওই জিনটা থেকে ওটা রিমুভ হতে হবে তখন?
Actually, they can say by direct observation. In many cases, the mechanism is known, and that is included in your syllabus. You should go through the histone modification chapter for those things. And in many cases, we don't understand the process properly. But result is, you can see that if you use a microscopy powerful enough to actually visualize those things, or fluorescent tag, you will be able to actually see that this histone has been removed from this cell and it is yet intact in other cells. Or if you use tagging, immunofluorescence technology, then you can also detect that the normal variant has been replaced by a separate cell variant. But how this entire process is not fully understood, but in many cases that is understood. For that, actually you should follow the this chapter. Otherwise, they demand actually separate class. Okay, so hopefully it's okay for you for this time being. পুরোটা হয়তো বুঝতে পারোনি, কিন্তু এইটুকু বুঝতে যদি পারো যে some because of some variant, because of removal, because of say কি বলবো sliding the histone through some portion, some DNA gets most accessible to say RNA poles and some not. এইটুকু বুঝতে পেরে থাকো, then I'm happy. বুঝতে পেরেছো তো কি এইটুকু?
হ্যাঁ স্যার।
ওকে, ঠিক আছে, থ্যাংক ইউ। এন্ড মানে আমার ভালো লাগলো তোমার মধ্যে কিউরিওসিটি ইনডিউসড হলো। হ্যাঁ, মেবি ইন দ্য নেক্সট সে ইন্ডাকশন প্রোগ্রাম, সেখানে হয়তো আমরা এটা নিয়ে আলোচনা করব, ইন্টার প্রসেস। ওকে, সো লেটস মুভ অন। So now the insulated gene, insulators. Now what are the insulators? They are again their proteins. They actually provide margin. We are not going to discuss them in detail. We don't have time for that. Okay, so what they do, they actually set a kind of limit so that a histone modification, some modification could occur. For example, say histone acetylase, we know that enzyme actually what it does, say the acetylase, they add acetyl group to the histones of the DNA, and this acetylation in some cases activates, in most cases activates the DNA transcription. In other cases, they may also inhibit the transcription. So this acetylation, what it does more or less, it changes the behavior of the DNA. Now, if you recruit, say for example, say a histone acetylase here, and leave it, then what it will do, it will move along the DNA and keep on acetyling everything that actually comes to their path. But you don't want that secret. You want to acetylate a particular region. Actually, not you. The cell wants to actually acetylate particular region of the DNA. And how to give limit to that? And this limit is actually given by these insulators. So in this case, you can see the insulator has been actually placed before and after this section of the DNA. Thus, the histone acetylase don't have any actually option other than to acetylase these middle regions. It will be recruited here and it will acetylate up to that position, then it will leave the promoter. So it will be able to acetylate specific region of the DNA and then activate specific or repress specific region of the DNA. Also, actually, they insulate one gene from the other. For example, say this is the enhancer and this is the promoter of gene A, and you have a given signal to this regulatory say the transcription factor that actually bound to the enhancer, and they activated the promoter, and now this gene is being expressed at enhanced state. অনেক দ্রুত গতিতে এখানে এটা হচ্ছে। Now you don't want the gene B to be expressed as fast as gene 1 or gene A. Gene A এর সমান তুমি as fast চাও না, If you don't place a say insulator, in many cases, single enhancer may actually regulate transcription of a number of genes. অনেকগুলো gene কে ওরা regulate করতে পারে একটা enhancer. So if you don't want to actually activate gene B, then you have to place again insulator. So what insulators does, it insulates gene A from gene B. So it will be expressed normally, whereas this one will be expressed at a faster rate. Okay. Let's move on to the next slide. So clearing this one. Hopefully you got the point. বুঝতে পেরেছো আশা করা যায়। 1 minute please, একটা ফোন আসছে বারবার আমার। So sorry about that, and hopefully you got the point what insulator does, and that you actually need to know. I know lots of question has been actually induced in your mind, and that's a good thing. But if you got the basic point, then I am okay. Okay. So can I move to the next point, or should I re-explain it, or do you have any problem? যদি থাকে আমাকে বলবে অথবা আমি সামনে যাব। Okay, so I think you don't have any questions.
Sir,
বলো তুমি
Sir,
কে
please.
বলছো কোথা থেকে বলছো?
sir, I'm from Kharagpur College, Somodip Samanta.
হ্যাঁ
Can you please re-explain it?
Do you explain what the function of insulators, or
The function,
oh.
only the function.
So actually insulator, actually in short, if you want to know what actually insulator does, then I have to actually say they act as barrier. So what they do, they act as barrier. So I'm writing it, and if possible, you should also write it acts as a barrier. A barrier for what? Barrier for chromatin remodeling complexes. For example, histone modifier, histone acetylase or something. For example, chromatin modifying complexes. For example, histone acetylase. And in this case, that function has been shown. In this case, if you actually recruit these chromatin modifying complexes, for example, histone acetylase, then what they will do, it will start acetyling and keep on acetyling, and you don't want that. You want to actually acetylate a particular region, and which region you specify using this insulator. So you have placed one insulator before and one insulator after. So now these complexes will have the access to this region only. So first thing, they act as barrier to what? Chromosome modifying complexes, histone acetylase, deacetylase, and other say DAM methylase and others, histone modifying complexes, they act as barrier. ঠিক আছে, এটুকু বুঝতে পেরেছো barrierটা?
Yes, sir.
Okay. Now, what the second function? Second function is they acts as insulator, separating one gene from the other from the effect of say promoters, say enhancers. For example, one enhancer could actually regulate rate of expression of more than one gene A, B, C, and D. So if enhancer is deployed here, now what enhancer are? We know they are regulatory transcription factor. When they will bind to enhancers, then what will happen? The rate of the transcription of all these genes will increase. But you don't want that. For example, you want to actually enhance the rate of gene A only, and you want to separate B and C. How do we separate? The cell actually separate by deploying again insulators. So they will place insulator just before the beginning of the gene B. So effect of the enhancer will be actually effect only the gene A, and B and G, the C gene will be insulated from the effect of enhancer. So they do two things. Both of this, they act as barriers. In first case, they act as barrier for histone modifying complexes and allow modification of certain region or specific regions of DNA. And in other cases, they act barrier to the enhancers and allow the enhancer to actually regulate gene expression of certain genes and don't allow expression of others. So is it clear now?
Yes, sir.
Okay, thank you. So now let's move on to the next slide. And the next slide is totally different thing. And that is the difference, the definition of genotype. So please keep that understanding what you have learned from the previous discussion aside, and again actually consider a totally different thing, make it a blank, and let's start learning a totally different thing, and later on we shall actually combine both of the two. Now we are actually concentrating on this thing, that is the definition of genes, and now I am not going to give you the definition. I want the definition from you. Any one of you actually, would you please actually give me the definition? What do you mean by no? So not genotype, it should be phenotype. Okay, say PHENO. It's a wrong, big mistake. What is the definition of phenotype? So please define phenotype. Not this. Okay, কেউ একজন বলো দেখি, কেউ তোমাদের পারা উচিত এইচএস থেকে জিনোটাইপ ফিনোটাইপ করছো, কেউ একজন বলো।
Sir.
হ্যাঁ বলো, তুমি কে বলছো, কোন কলেজ থেকে বলছো?
Sir, Shriparna Chattopadhyay from Ashutosh College. Sir, phenotype is the observable characters or the expression of the genotype, rather. The expression of the genotype in which we can see the characters inbuilt in an organism.
Okay, fine. Anyone other than Shriparna? হ্যাঁ বলো, তুমি কে বলছো?
So myself Sangeeta Lala from Kalna College.
Okay.
So the phenotype is the morphological characteristic. That's why we can just distinguish any species from another. And the genotype is responsible for creating the phenotype.
And genotype is the responsible for creating the phenotype. Or expression of genotype is the phenotype. Fine, yes, fine, very good, very good. Any one of you? Anyone other than this?
Sir,
বলো, তুমি কে বলছো, কোন কলেজ?
Sir, I am Shubhodip Poddar from St. Xavier's College.
Okay.
Sir, phenotype can be defined as the observable characteristic features that arises from the interaction between the genotype and the environment.
Exactly. হ্যাঁ বলো।
Yes, sir, that's all.
Thank you. তোমার নাম কি বললে গো?
Sir, Shubhodip Poddar.
Shubhodip, thank you Shubhodip for giving me the answer. That is the answer. And what is it? Let's have a look at this. So we have the definition written here, and this is from the book Stanfield Genetics. Now if you have the opportunity, please grab that book and start actually studying that book immediately. It's a very good book, and this will help you to actually develop your analytical skills. Okay, so what actually definition means, and it is written here, any measurable characteristics we can actually measure, or distinctive trait. For example, you can count or you can measure weight, body weight, or say number of fingers or number of feathers, scales, etc. Any observable characteristics or distinctive trait, for example, color or anything else, or the trait that is not even visible to naked eye, requires special tests. For example, ABO blood group. These things, this is the main line. Phenotype is the genotype brought to expression in a special set of environmental condition. That you actually told me. Actually, genotype, if you express it, it will give the phenotype. Okay, no problem. Up to definition fine, definition, but you forgot to mention the environmental condition. So genotype, expression equal to phenotype. But again, there is a term environment. Why I am stressing the term environment? The previous definitions are true and they are good definitions as well. But why we are giving in the term giving stress on the term environment? Because actually in many cases same genotype same, sorry about, if there is a spelling mistake, then be sure that it's me. Okay, a same genotype can have more than one phenotype. Okay, so same genotype, for example, how could it produce more than one phenotype? Do we have any example? Yes, we have. And that example is this Himalayan say phenotype. Now Himalayan phenotype is actually regulated by 3 genes. It's a multiple allele, controlled by multiple alleles. 1 gene, multiple alleles, 3 alleles. These are actually 4 alleles: C, CH, CH, CCH, CH, and C. So we have 4 alleles for the same gene, and we know that one individual can carry only 2. So one individual can have say combination of these 3 genes. For example, one could have CC or CH, capital CCH, or capital C CH, or capital C small c for C allele, and other combinations as well. There will be lots of combinations. So any 2, যে কোনো দুটো hold করতে পারে, because we are diploid and we have 2 chromosomes and we have 2 locus to hold that gene, and that's why we can hold only 2 genes, and this lead to actually 4 type of phenotypes, and which are if the individual is C dash means 1 capital C, and in the dash it may be CH, CCH, CH, or C. That is actually meant by dash. If the individual is C dash, then individual will have full color. What do we mean by that? This one is the full color. So capital C, or capital C dash. Capital C dash will do the same. C dash means any 4 of these alleles. So we'll have full colors. It's grayish brown color setup. Now, if individual have this say genetic complement, that is CCH, CCH, or CCHC, then we'll have a color which is silvery white, which is known as chinchilla. Now, why? Because they have this type of dominance relationship. We don't have the opportunity to discuss dominant relationship multiple allele. They deserve say separate class on their own right. But for understanding purpose, let's actually think that if it is this one and then that one, we cannot have the entire idea. But let's that if it is have CH, CH, and CH, then it will produce Himalayan phenotype with say darker extremities. You can see the older region of the body. It becomes black, and if it is small c, then it will produce the all white. So if we look at this genotype, then we can see that combination of different allele would produce different phenotypes. Now, if we actually look at this genotype only, and to actually remove any type of controversy, let us consider we are discussing this gene only. Okay, we are not discussing this gene. So what should be the phenotype of this if some individual would carry this one? যদি একটা ইন্ডিভিজুয়ালের CH তোমার জিনোটাইপ হবে থাকে, তার ফিনোটাইপটা কি হবে? ফুল কালার, চিনচিলা, হিমালয়ান, না অ্যালবিনো? কে বলবে? অর্ক বলছিল, অর্কই বলে দাও। খুব ইজি কোশ্চেন।
সো কোশ্চেনটা একবার রিপিট করবেন?
বলছি যদি একটা ইন্ডিভিজুয়ালের জিনোটাইপ CCH হয়, মানে CCH সরি CHCH হয়, তাহলে তার ফিনোটাইপ কি হবে? বলো কেউ, না পারলে ধরে নেবো তোমরা পারছো না। সে কি একটা ইন্ডিভিজুয়ালের জিনোটাইপ এটা হলে তার ফিনোটাইপ কোনটা হবে? এটা হলে কোনটা হবে? হ্যাঁ, হিমালয়ান হবে। এটা তো খুব ইজি। So anyway, so it will have Himalayan phenotype, no problem. But there is one. So if we consider this one, so where is the problem of the first definition? So if we have the CHCH, if we bring this to actually expression, it should produce this one, that is the Himalayan phenotype. That means a rabbit with say a black extremities. All cold regions will produce a black color. But this is not always the case. সব সময় যেটা হবে, it doesn't mean এটা কিন্তু নয়. Then all of them have this genotype. How it could produce more than one phenotype? Would it produce different phenotype? Yes, of course it will produce different phenotype. Again, it's wrong. Same genotype. Okay, some will also do. Some genotype can have, ঠিক আছে, খুব ভুল নেই, some দিয়েও চলবে। So কি করে same genotype, that is CHCH, can produce this one as well as this phenotype, all albino,
So
depending on
environmental conditions.
Yes, depending on environmental condition. If the temperature is low, below 15 degrees, normally it is 15 to 25 degrees centigrade. That is actually normally we refer. So if the temperature is low, then it will produce this phenotype, that is Himalayan phenotype. And if temperature is high, then it will produce this all albino. Now, how it is actually produced? This black color is produced by an enzyme that is temperature sensitive. If the temperature is high, then this enzyme will be degraded and will not work properly. So a wall of white, that is albino phenotype will produce. So you can see that in some cases, same genotype can produce two different phenotypes. So where is the problem? The problem is in the environment. So environment in this case, changing the expression of the gene. What do you mean by genetic expression? Gene expression, we learned that up to the point of protein function. If protein ke tumi modify karo, phosphorylate karo by other some means, for example, in this case we are actually giving heat, and because of this heat treatment, this protein is actually losing its function, so it is producing an all white say phenotype. So in this case, gene expression is being changed because of the say environment. Okay, so what we are going to learn actually today, this topic is environmental regulation of gene expression. And that's why actually I given the example, and why the last definition by Orko, I think, was better than the other two, because this definition also includes the environment, and you can see same gene while being expressed is actually giving the phenotype, but two different phenotype based on the environmental condition. So if you have any question up to this point, you can ask. And actually what you can do, you can actually induce black regions by actually say adding coal blacks to a specific region. No problem, add coal black there. And the coal black, what it will do, it will keep the so normally what happens, the core body temperature is always higher than the extreme. Ekhane batas lage, and heat actually radiates easily from the ears and nose and feet. That's why these regions are actually colder, and because of this colder temperature, the action of the say enzyme is retained, and as the core temperature is high, so it gets degraded and the region becomes albino. So if you want some portion of this rabbit to be say black, what you have to do, you have to just place an ice bag on that region, and that region the temperature will become low, and at the low temperature the enzyme will survive, and that region will become say black again. So by just changing temperature, you can regulate gene expression. Okay, so up to this point, if any of you have any question, you are again free to ask, otherwise I shall move forward. Aro thakle amake korte pare. Okay
স্যার আমার একটা
কি
কোশ্চেন
বলছো,
ছিল।
কোন কলেজ থেকে বলছো?
আমি স্যার দৈপায়ন ঘোষ ফ্রম গুরুদাস কলেজ।
ওকে,
বলছি স্যার, যদি ওই মানে ওই স্পেসিমেনটাকে যদি একটা কোনো ক্লোজড রুমে রাখা হয় যেখানে টেম্পারেচারটা বাড়িয়ে দেওয়া হলো, সেখানেও কি তাহলে ওর ওরকম কোট কালার চেঞ্জ হয়ে যাবে?
that should actually, it will take time, but as the enzyme activity will be lost, so subsequent hair follicles will not be actually able to produce blacks, say colored hair, and it will be ultimately become say all albino. Yes, everything is dependent on the temperature. So the temperature যদি change করো, it will change its expression. ঠিক আছে?
Okay sir, thank you sir.
Thank you. So let's move on. So what we have learned from this discussion, we have learned that actually say one gene can have multiple phenotypes. That is, one gene have the ability to produce more than one phenotype. The ability to produce more than one phenotype, and this phenomenon is known as phenotypic plasticity. Okay, so we define this term. So same CH genotype has the ability to actually, if CH is enough, so actually has the ability to produce two phenotype. One is Himalayan, and another is say albino. So one genotype has the ability to produce more than one phenotype. Based on different environmental condition or some other conditions, is known as phenotypic plasticity. Okay. So we are actually discussing developmental biology. So when such plasticity involves actually associated with say related with development, then we call it say developmental plasticity. Okay, acha. And actually, plasticity could be two types. One is reaction norm, and is second one is polyphenism. And don't have scope to actually discuss them. But reaction norm, when the effect is continuous, it is called reaction norm, and when the reaction is say discontinuous, it is called polyphenism. In general, a little bit oversimplification, but it will do. So what do you mean by say when it is continuous? We have now. So when it is continuous. And what do we mean by continuous? Continuous ko katan mani. And what do we mean by discontinuous? Discontinuous phenotype. Phenotype continuous, discontinuous. What is this? This is when the effect or one effect is actually very difficult to distinguish from another. Based on condition. For example, height of human is a continuous character, and there is multitude of possibility in between. For example, human height may vary from say 2 feet in case of dwarfs to say maybe 7.5 or 8 feet. Let's consider it 8 feet, and you can have any possible values between them. Say 2.3, 7.99. 0.7.99999, anything, any value up to infinity. When any character or anything, any variable has this type of trait, we call it a continuous variable. In this case, we are calling it a continuous phenotype. So when phenotypic plasticity produces phenotype, that can be anything, that can have any value in between two extremes. We call it say continuous. Jodi সম্ভবত উদাহরণ দিলে it would have been better, but at this point I cannot recall one. একটা মনে করতে পারছি না. Anyway, আমি ভেবেছিলাম আলোচনা করব না, করে ফেললাম. Now, what do you mean by continuous? Say discontinuous. Discontinuous means you can actually distinguish or segregate the different phenotypes. For example, in case of Himalayan, you can easily segregate the Himalayan one with albino one. Himalayan one has black extremities, whereas albino one is totally white. So we have a binary type or binary tertiary, quaternary, whatever may be the case. The cases are distinct from one another. You cannot expect any intermediate values. Okay, when such type of event occurs, that type of variable is called as discontinuous variable. In this case, we are calling it discontinuous say phenotypic plasticity, and this discontinuous phenotypic plasticity should be known as polyphenism. Example, Himalayan rabbit, we have already have one. So have you, any of you have any problem up to this point? If you have, you are always free to ask. Otherwise, I shall move to the next slide. So hopefully you don't have. Don't feel shy. Please ask. যত বোকা বোকাই প্রশ্ন হোক না কেন করবে, হ্যাঁ, লজ্জা পাবে। Okay, now. Now, what else other than environment can modify the phenotype of a particular genotype? So, up to this point, we have learned that temperature, there is environment have some role that can modify the phenotype. What else other than say environment could actually lead to phenotypic plasticity? These are non-Mendelian inheritance. For example, maternal effect could actually change the say cell coiling of Lemnia peregrina, epigenetic inheritance, cytoplasmic inheritance. All these things actually may lead to phenotypic plasticity, as well as extended genome. What do you mean by extended genome? These are the symbiotes. Previously, we thought that our genome means what makes us is our nuclear DNA. That is the only thing need to make me or you. আমাদের নিউক্লিয়ার জিনোম যা আছে সেটাই যথেষ্ট, ঠিক আছে? But later on we found that this is not true. We have a mitochondrial DNA as well. So mitochondrial DNA as well, DNA from chloroplast as well. It's the cytoplasmic inheritance. That also have significant effect. Some diseases are actually associated with this defect in mitochondrial DNA. So we have a mitochondrial DNA as well, which makes me and you. That makes us. So not only nuclear DNA, we have mitochondrial DNA as well, but they also actually reside inside our cells. So that is ours, no problem. But later on, particularly in the recent decades, scientists have found that actually not only the nuclear genome or the mitochondrial DNA actually something that makes me or you is actually dependent on our symbiotic bacteria that lives on our skin, our gut. They actually give us different ability to absorb different nutrient and thus ability to survive at different conditions. So same condition, because of the presence of different gut bacteria in my gut, will allow me to survive or allow me to perish and allow somebody else to actually survive. So this genome of symbiotic bacteria could also lead to actually survival and phenotype plasticity. So this is known as extended genome. What is it? It is actually symbiont of bacteria. So all the gut bacteria, all the bacteria that live in our body, that also actually should be included in our genome, as they are not present inside our cell and as they are not transferred through say reproduction, normal process of inheritance, they are called extended genome. But you have to remember that they also say help in phenotypic plasticity. Okay, so we have learned what else other than environment could actually play a role in case of say production of phenotypic say plasticity. Now we shall actually focus on our main subject, that is temperature dependent regulation of gene expression and example. And what is the example? The example is eye spot formation in the wings of Malawian butterfly Bicyclus anina. Okay, so what is it? It's a butterfly, of course, you can see, and the name also suggests that it's a butterfly. And what we are going to study, what polymorphism we are going to study? We are going to study the eye spot formation. Hopefully you can see in this picture on the right hand side that has say aqua background has been actually marked wet season morph, has large eye spot on their wing. So you can see that this is okay. Let's use another color. Maybe you will do better job here. No, it's not. So let's use black. Okay, so you can see that in this level it has eye spot in their wing, but the dry season morph has eye spot anyway, but they are less pronounced. Now how this eye spot forms in wet season and in dry season, or alternatively we can ask how the expression of this eye spot is regulated in dry and wet seasons. Now we are actually interested in discussing the regulation of eye spot formation in different seasons. How it is actually regulated? It is regulated by actually temperature. Okay, 1 minute, please. So let's move on to the next topic, and that is how this eye spot the regulation is generated. Actually, dry morph is produced in winter season. So winter season experiences very less amount of say rainfall, and it's dry, and it is because of this dry season actually the leaves fall on the ground, and the absence of eye spot actually helps the butterfly to blend in, and it occurs in low temperature. So the dry morph also you can call it low temperature morph. And the wet season morph is, we can also call it say high temperature morph. High temperature morph. Okay. Or morph with eye spot, or with eye spot and without eye spot. And you can actually call. So so you know the conditions. Which one is dry season? Which one is wet season? If you are confusing, you should write them up, because if you don't remember this actually memory thing, it had a memory factor thing. If you cannot recall, then later on it may actually make you confused. Konta dry, konta wet, konta শীতকালে হচ্ছে, konta গ্রীষ্মকালে হচ্ছে. So this dry one is in the winter, that is in low temperature. It is in summer and high temperature. So this is it. So how actually this difference in gene expression is occurring? When it actually, it of course occurs during the development. Actually, development of wing actually starts from, imagine, on every organ. In case of mammals, it occurs from organ rudiments. We call it organ rudiments. For example, lung develops from lung rudiment, and liver from liver rudiment, and sort of things. And in case of say insect, they develop from discs known as imaginal discs. And in case of wing actually develops from wing imaginal discs. And now forgive me, it is a picture of Drosophila. not the butterfly. It was not available in the internet. I have done it very quickly, so sorry about that. So anything, it will give you some impression. So wings will be developed from wing imaginal disk. So this regulation occurs in wing imaginal disk. In the wing imaginal disk, the expression of distal-less gene is the important. Gene is important. If distal-less gene is active, then it will convert wing imaginal disk in such a way that it will produce eye spot. That means expression. We have distal-less genes, DLL. So for short, use DLL. So we have DLL gene, and if it get expressed, then it will produce DLL RNA, mRNA, and mRNA will finally produce DLL protein, distal-less protein. And if distal-less protein is present, then it will actually produce the eye spot. And if distal-less protein is not expressed, then there will be no mRNA, there will be no expression of DLL protein, and there will be no formation of eye spot. That's the eye spot will not form. Hopefully you got the point. And if you have any confusion understanding this, because it's vital for understanding, you are always free to ask. Turn on your mic and please ask. I'm giving you 1 or 2 seconds for that. Okay, so I will assume that all of you understood it. So everything depends on expression of distal-less genes, and when the distal-less gene will be expressed. Answer is when there will be a high concentration of 20-hydroxy ecdysone 20E. So high 20E concentration will activate distal-less gene and will actually start to form eye spot, will form the eye spot. Now, when there will be high 20 concentration, the answer is high temperature. When the temperature will be high, high temperature will help to accumulate 20E, that is 20 hydroxy say ecdysone, and a high 20E excitation concentration will lead to activation of distal-less gene, and this activation of distal-less gene will lead to formation of eye spot. If actually temperature is low, then what will happen? Let's use another color, maybe the violet one. So now temperature is low. So when the temperature is low, it will not allow say accumulation of 20E. So 20E concentration will be very low. Now, when the 20E concentration will be very low, the DLL gene will not be expressed. And DLL gene, as it will not be expressed, the eye spot will not form. Okay, so it's easy. I think it's easy, and I think all of you got the point. Now, if you have any problem, you are always free to ask. Unmute yourself and ask. যদি প্রশ্ন থাকে, নাহলে আমি পরেরটাতে মুভ করব। আছে?
অ্যাডাল্টে যদি ওই জিনটা মিউটেশন করে দেয় তাহলেও কি ওরকম হবে?
না, মিউটেশন হলে তো সেটা ডিফারেন্ট, তাহলে তারা সেম জিন হলো না। Mutation will produce a different allele. Same gene maybe, but it will produce different allele. So we are not considering that. We are considering same allele as well. Same gene is same thing, but different expression. Okay.
Okay, sir. Thank you.
So the mutation is a different story. So mutation may lead to larger eye spot or smaller eye spot, but we are not considering that. We are considering same gene. Okay. Same gene, but their expression, difference in expression. So how this is achieved? How this difference in expression is achieved? Now let us move to the next slide. Oh, okay, we have discussed it already. Where from they are produced? Yes, they are produced from prosthoracic gland, ecdysone, and then ecdysone is converted into 20E in the body tissue, and actually ecdysone is released from the brain through the action of PTTH. PTTH tomorrow release for that's the process. You should be interested, but not absolutely necessary. Now, thank you. Now, another thing, actually, the slide policies are not good only because actually I'm outstation. I forgot to copy that. For you, I actually had created one animation as well, but unfortunately, I forgot to copy that. So anyway, what you have to do, you have to just view the concept from the images. In the image I have, so I'm stopping presenting this one and actually presenting the image. 1 minute, please. How that actually the regulation occurs. So I'm going going to discard all of these things. And now again, I am presenting the picture. The schematic diagram, and it should help you to actually understand. So let's try to present it. So I'm totally new in Zoom and don't know how to present that. So is it being presented? একটা ছবি কি তোমরা দেখতে পাচ্ছ? কেউ একজন আনমিউট করে বলো।
schematic ওটা দেখতে পাচ্ছি স্যার with the brain and
oh no, that no, then I have to stop and represent screen. This one. Okay, so hopefully it is visible now. Is it visible?
Yes sir, yes sir, temperature dependent.
Oh, okay, temperature dependent regulation of gene expressions in bisectors and anemia. আমার খুব কষ্ট হচ্ছে, আমি কষ্ট করে জানো তো বানালাম তোমাদের জন্য এটা অ্যানিমেশনটা, but I forgot. বিরক্ত লাগে বলে তো। Anyway, so let's discuss them side by side. So what will happen if the temperature is low, and what will happen if temperature is high, and how the gene expression will be regulated. So let's consider left hand side we have low temperature, and on the right hand side we have high temperature, and it is already marked. And for your better understanding, I'm using a pen to mark it anyway. So this side is a low temperature, and this side is a high temperature. As I have mentioned, low temperature will not allow the accumulation of 20-hydroxy excitation. That means it will have very low concentration outside of the cell. So if the temperature is high, then high temperature will actually allow the accumulation of 20-hydroxy excitation, and their concentration will be high. It is easy, and it has been actually represented here. And now let's move on. And this is actually the plasma membrane. বলো, কেউ কিছু বলছো? কেউ কি কিছু বলছো বলো? So this one is the plasma membrane. So 20-hydroxy say estradiol, ecdysone is a steroid hormone, and we know that all steroid hormone actually shares a nucleus which is known as cyclopentanoparhydrophenanthrin nucleus, and which is derived from the cholesterol and is hydrophobic in nature. So they are hydrophobic. Main thing is they're hydrophobic. All the steroid hormones. So they have the ability to pass, as they're hydrophobic, they can freely pass the plasma membrane. So this will also be able to pass the plasma membrane, and that too. And what they will do after entering the cytoplasm, they will bind to their receptors. All steroid hormones have cytoplasmic or nuclear receptors. So they will bind to their receptors, and they are located in the cytoplasm. So in this case as well, they will bind to their receptor, which is known as ECR. Okay, so in that case as well, it will bind to their receptor ECR. But as it has high concentration, it will bind to an additional receptor that is also a receptor for 20, known as UPS. And this binding, like all other steroid hormone signaling or thyroid hormone signaling, will lead to dimerization. And বলেছিলাম the dimer form করবে. And in this case, as the concentration is low, very few molecules will bind to both of the receptor, and fewer will form dimer. And after dimer formation, let's move on top. And after dimer formation, they will enter inside the nucleus. Here, no dimer has formed, so no one will enter the nucleus. And the nucleus, the enhancer for distal less gene. So this one is the distal less gene. The enhancer for distal less gene will be, this is the promoter for distal less gene, this region. Okay, it's not marking, so let's mark it here. So this is the promoter. This is the promoter for this distal less gene, where the RNA pol will bind and start the transcription. We have learned it from the previous discussion. And this is the enhancer to which the actually regulatory transcription factors will bind. So as no receptor complex, this is the actually regulatory transcription factor. So as there is no regulatory transcription factor, the enhancer will remain bound to a inhibitor, and that will allow binding of another repressor called E75. And these two will repress the promoter, and there will be no transcription or no DLL protein. And as there will be no DLL mRNA, so there will be no DLL protein. As there will be no DLL protein, there will be no I-spot. So why actually I-spot don't from, sorry, let me try to actually scroll it, but somehow it's not happening. Okay. So now, so you can see that, so as there is no transcription, no DLL protein, so there will be no I-spot formation. That's how actually temperature regulate actually inhibits the expression. So normally it remains in the inhibited state. Now, how actually high temperature leads to its activation? Now we have seen that high temperature means elevated level of say 20E, and elevated 20E means formation of this receptor dimer, and this receptor dimer will then replace the inhibitor from the enhancer. So the inhibitor got replaced and will competitively also remove say the E75. And as both of these will be actually removed from the enhancer, because the say receptor dimer will get bound to this this enhancer. And I have already mentioned এর আগে যখন পড়িয়েছিলাম যে তখন তোমাদের একটু বললাম দেখো, if you can recall that these are the regulatory transcription factors, that what they do, they actually bind to the enhancers or regulatory sequences and thereby activate genes or repress gene transcription. So in this case, what they are doing, they are actually recruiting a histone methyl transferase that is activating the promoter and allowing the RNA pol to bound to it and thereby initiating the transcription. And once the transcription is on, it will actually produce DLL protein, and once DLL protein is present, it will activate downstream genes and will lead to formation of I-spot. Okay, so hopefully you got the point. If any of you have any question, you're always free to ask from first to last point. আরো কিছু আছে, বুঝতে কোনো অসুবিধা আছে এই জায়গাটা? আছে কি? না থাকলে না বলে দাও, তাহলে আমি এটাকে শেষ করব।
ওকে স্যার।
ওকে, থ্যাংক ইউ।
বলছি এই রিসেপ্টর ডাইমার যদি না ফর্ম হয় তাহলে কোনো কাজ হবে না না?
না, তোমাদের বললাম যে actually gene transcription is being regulated by regulation term is actually dependent on the regulatory transcription factor. If you don't have regulatory transcription factor, how would you regulate the gene transcription? যদি এটা না থাকে, ওটাই তো regulatory, that is actually regulating the rate of transcription. In this case it is absent, that's why it is not being say transcribed. In this case it's present, and that's why it is being transcribed. So if no receptor dimer, that is actually the regulatory transcription factor, no transcription. Okay, বুঝতে পেরেছো? Okay, thank you for asking questions. So
স্যার,
হ্যাঁ বলো।
স্যার বলছিলাম যে হিস্টোন মানে যে একটা কনসেপ্ট ছিল না যে ইউক্রোমাটিনাইজেশন হেটেরোক্রোমাটিনাইজেশন, তো ওখানে দেখেছিলাম যে মিথাইলেশন হলে তো অ্যাকচুয়ালি ওই মানে কি বলবো, মানে মিথাইলেশন হলে তো ইনঅ্যাক্টিভ হয়, তো এখানে
normally these are actually case specific, no?
আচ্ছা আচ্ছা।
Normally acetylation means activation and methylation means deactivation, huh? But they are not always the case. আমি যখন প্রথমে বললাম at the beginning of our lecture, I actually mentioned normally it's true but not always. In this case you can see histone methyl transferase is actually leading to the activation of say transcription in this case, in particular in this case. Okay, so for that you can go through Albert's Albert's একটা সেকশন আছে, দেখবে histones and its modification, তাতে দেখবে কতগুলো সাইট রয়েছে, where they should be methylated, acetylated, phosphorylated to form what, to give what result. It's beautifully explained in that, so we should actually follow that actual topic. Okay,
okay sir, thank you sir.
Okay, so I'm stopping stopping sharing this.
Sir.
বলো।
Sir, I have a question. Sir, in a very broad perspective, so apart from the Himalayan rabbits and the butterfly examples we saw, the phenomena of this phenotypic plasticity, is it found in other animal models too?
Yes, like, of course it's found, and actually if you go through your developmental biology book, then we'll find it is observed in locusts, it is observed in say caterpillars, it is observed in say mice, it is observed in beetles, and there are lots of examples, and if you are interested you should Google it and you will get lots of thousands of examples of phenotypic plasticity.
Sir, মানে এরকম হতে পারে if we can relate any disease condition with that of the, like it is also a phenotype, যদি কোনো genotypic changes হয় বা
Of course, we have phenotypic plasticity and the effect of environment, and that is actually alkaptonuria or alpha ketonuria. Okay, alpha-ketonuria, why it happens? Because of the presence of phenylalanine and conversion of phenylalanine into disease-causing agent. I forgot the exact chemical, but if phenylalanine is present in say diet, it will lead to say disease condition. But if somebody is raised from their initial period in food that is divided of phenylalanine or absent from phenylalanine, you will have normal good phenotype. So we can relate phenotypic plasticity with disease as well. And there are lots of other examples. At this moment, I can recall this one, and there are better say example out there. Anything else? Are you now, thanks for participating. Now it's I'm actually sending it.
No, actually, I was telling
to
that
Shoma.
You discussed. Yes, sir. I'm Shoma, and I just want to say that you discussed this topic in such a very interactive way. I think participants are very much enriched by your talk, and I would like to thank you as well as the participants who have made this session very live and interactive. Actually, we want this kind of participants also because the program is for the students or the new aspirants who are willing to do something creative, new, and who are very inquisitive mindedness. Thank you all the participants, and thank you, Dr. Rahman. It was very nice presentation.
Thank you.
I'm very thankful that you have given us so much time, and you are just amazing. Thank you. And
now that's a overstatement actually.
The question answer session is open officially from now, but I think we had a lot of questions during your talk, but I would like to take some questions that are in our chat box, and after that we will continue the interactive sessions for a few minutes also, where the participants can unmute themselves and ask the questions themselves. Okay, so we are taking some few questions that are in the chat box. In our first question in the chat box is, sir, how the rate of transcription slows through condensing of DNA?
So would you please mind to repeat it?
How the rate of transcription slow through condensing of DNA?
Now, I have actually already mentioned in my presentation that actually normally DNA, eukaryotic DNA in particular, they don't remain free and easily accessible to the RNA polymerase and other transcription factors. To actually gain access, you need to actually free that DNA. And if this freeing is actually done by acetylation and other means, and if you keep it in highly condensed state, then it's very easy. The DNA will not be accessible to the RNA polymerases and others enzyme, and it will not be expressed. That's how this condensation itself is not actually allowing the transcription rate to occur, because it is not allowing the enzymes to access the DNA freely. Okay.
Okay, thank you. The next question is from Abhishek Das. How can environmental factors affect the expression of genes Quizlet?
Genes?
How can environmental factors genes Quizlet?
Okay,
genes Quizlet.
I actually, it is not possible for me to actually make out what he actually meant. Gene Quizlet, I don't have any idea. Actually, I'm sorry.
By muting.
I don't have any idea how environment regulates those. পরেরটা শোনা গেল না, সোমা প্লিজ।
First question is what types of environmental factors can influence DNA methylation?
Okay, actually, if this type of answer I have to be specific, you know, and for that I have to go through papers in which specific genes and methylation and deactivation and has been gone through. And for your question, when methylation occurs, when methylation does not occur, actually it will depend on different environmental conditions, and they will activate different set of circumstances and will lead to different methylation or acetylation. And to give specific example, I have to study specific example and give you answer. At this present, actually, I don't have any answer where say specific factors, how they activate methylation. For that, I will suggest you to go through Google Scholar and give a search, and that will give you a better answer. Okay. In this case, we have seen that in case of Malawian butterfly, that it recruits say methylase, histone methyl transferase. So you can actually assume that in this case methylation activates, but when it suppresses, we have to actually go through separate study. এমনি মুখে বলা যাবে না, separate study করে বলা যাবে, বা তোমরা যদি মনে করো, you can always Google it.
Okay, sir. We can see that there are lots of thanking messages telling that your session was very helpful for the participants. They have written.
Thank you.
In the
Thank you. You're most welcome.
Also,
I am honored actually.
they are very much enjoyable session actually. And now I would like to request our participants, if they have some more questions, they can mute themselves and ask the question. If anyone have any question. Shamanto, you unmute yourself and ask the question. Or should I read? He has told that, he has asked that, sir, as you told, DLL genes express the eye spots in Malawian butterfly. In dry season, the DLL genes are inactive. Still, there are spots not prominent. Why is it?
Okay, please, if it was possible for you, please unmute and ask the question. It is actually difficult to, okay, let me check the chat box, how to check that. So if it is possible for you, please ask. হ্যাঁ বলো জিজ্ঞেস করো কি বলছে।
সৌম্যদীপ, তুমি বলো, আনমিউট করে বলো।
হ্যাঁ, মানে ওই শুনে একবারে বোঝা যাচ্ছে না, তুমি বলো কি বলতে চাইছো। রিটেনটা দেখলে বোঝা যেত। এই তো চ্যাট।
He's telling that he has some problems in his microphone.
I'm going through his message. Okay. Sir, as you told, DLL gene expresses the eye spot in Malawian butterfly. Okay. In dry season, the DLL genes are inactive. Still, there are eye spot not prominent. Why is it? Okay. Interesting question. Actually, for prominent eye spot formation, actually expression is required. They actually produce a paracrine factor. This paracrine factor diffuses from the center, and as the amount of paracrine factor increases, the eye spot actually eventually forms through gradient of say paracrine factor. Actually, off means actually totally off. Not that means. Something, some background level of expression might be there, and that may cause the actually eye spot formation to look like that. Okay. For precise answer, you should go through the relevant studies over say Google Meet. Okay. Google Scholar just precise answer ache sekhane dekhbe. You should give search eye spot formation in Malawian butterfly, then we'll have some idea. Okay. These are not easy thing to answer in 1 or 2 words. You should cite specific say experiment and specific evidences on the basis of this basis, and these are experimental level questions. So For general purpose, this is my answer. But if you want to be sure, you should always search the literature. Okay. আচ্ছা আর কেউ কিছু বলবে?
I think participants actually they have asked so much questions during your lecture.
Okay.
Okay, thank you, Dr. Rahman. It was very nice to hear from you, and we hope that in future we would also be honored if you join
No,
us.
thank
in
you.
some
Thank you.
lecture.
Honor is mine. Honor is mine. Okay,
And,
thank you.
Thank you. And I thank all the participants as well who are listening to us and the organizing committee of our DBT Star College program, our principal sir, Dr. Shakon Kumar Mishra, Dr. Bidhan Chandra Samanto, who is the coordinator of this program, Dr. Kalipada Maiti, who is the joint coordinator of this program, and all the teaching and non-teaching staffs of our my college. And thank you, Dr. Rahman. We are concluding the session now. Okay, thank you so much.
Thank you. Thank you. So I'm leaving. Okay. Can I
Okay,
leave?
okay. Thank you.
Okay. Thank you.
Yes, yes. The session is over. Everyone can leave now. Thank you.